Bachelor of Science Nursing Microbiology. Unit 2, Growth and Nutrition of Microbes. Includes temperature, moisture, blood and body fluids influence the growth. Microbial growth refers to an increase in the number of cells rather than their size. The growth of microorganisms is influenced by various physical or environmental factors, chemical or nutritional factors, including temperature, moisture, nutrients, blood and body fluids. Factors affecting microbial growth. The growth of microorganisms is influenced by physical or environmental factors and chemical or nutritional factors. These factors determine their survival, reproduction, and metabolic activity. 1. Physical or environmental factors. These factors include conditions that directly affect microbial growth and metabolism. Temperature. Temperature plays a vital role in microbial growth, as different microorganisms thrive at specific temperature ranges. Psychrophiles, cold-loving microbes, grow between -5 degrees Celsius to 20 degrees Celsius, e.g. Pseudomonas species. Mesophiles, moderate temperature microbes, grow between 20 degrees Celsius to 45 degrees Celsius, including many human pathogens, e.g. Escherichia coli. Thermophiles, heat-loving microbes, grow between 45 degrees Celsius to 80 degrees Celsius, e.g. Bacillus stearothermophilus. Hyperthermophiles, extreme heat-loving microbes, grow above 80 degrees Celsius, e.g. Thermococcus species. High temperatures can denature microbial proteins, while low temperatures slow down metabolic activity, preventing growth. B. Moisture, water availability. Water is essential for microbial metabolism and growth. Microbes require water for enzyme activity, nutrient transport, and cell division. Desiccation, drying, inhibits microbial growth, but some bacteria form endospores, e.g. Clostridium botulinum, which allow them to survive in dry conditions. Osmotic pressure influences microbial survival. Halophiles, e.g. Halobacterium, thrive in high-salt environments. Osmotolerant microbes can survive in low water conditions, .C. pH, acidity or alkalinity. Microorganisms have an optimal pH range for growth. Example, acidophiles thrive in acidic conditions, e.g. Lactobacillus acidophilus in yogurt, Thiobacillus ferrooxidans in acid mine drainage. Neurophiles grow best at neutral pH, e.g. Staphylococcus aureus. Alkaliphiles survive in alkaline environments, e.g. Bacillus alkalophilus in soda lakes. D. Oxygen availability. Different microbes have varying oxygen requirements. Example, obligate aerobes require oxygen, e.g. Mycobacterium tuberculosis. Obligate anaerobes cannot tolerate oxygen, e.g. Clostridium botulinum. Facultative anaerobes grow with or without oxygen, e.g. E. coli. Microaerophiles require low oxygen levels, e.g. Helicobacter pylori in the stomach. E. Radiation and UV light. Some microbes are resistant to radiation, while others are destroyed by it. Example, Deinococcus radiodurans can withstand high radiation levels. Most bacteria are killed by UV radiation, which is used for sterilization. 2. Chemical or nutritional factors. Microorganisms require specific nutrients for survival and energy production. A. Carbon source microbes obtain carbon from organic or inorganic sources. Example, autotrophs use CO subscript 2 as a carbon source and i.e. cyanobacteria in photosynthesis. Heterotrophs obtain carbon from organic compounds, e.g. E. coli using glucose. B. Nitrogen, sulfur and phosphorus essential for protein, nucleic acid and ATP synthesis. Example, nitrogen-fixing bacteria convert atmospheric nitrogen into usable forms, e.g. rhizobium in legume roots. Sulfur bacteria use sulfur for energy, e.g. thiobacillus in sulfur springs. Phosphorus is required for DNA and ATP, e.g. Pseudomonas utilizes phosphate for growth. C. Trace elements, minerals. Microorganisms need small amounts of minerals for enzyme activity. Example, iron is essential for electron transport, e.g. Mycobacterium tuberculosis requires iron for survival. Magnesium and zinc are enzyme cofactors, e.g. Bacillus species require magnesium for sporulation. D. Growth factors. Some microbes require additional organic compounds like vitamins and amino acids. Example, lactic acid bacteria need vitamins for growth in dairy fermentation. Fastidious bacteria, such as Haemophilus influenzae, require Heme and NAD+ from their environment, AA, blood and body fluids. Microorganisms can use blood and body fluids as nutrient sources, which directly influence their growth. Blood supports the growth of pathogenic bacteria, especially those that require iron, e.g. Staphylococcus aureus and Neisseria meningitidis. Plasma and serum, rich in proteins and electrolytes, providing an ideal medium for bacterial and viral replication. Cerebrospinal fluid, CSF. Infections like meningitis occur when bacteria invade and multiply in the CSF, e.g. Streptococcus pneumoniae. Saliva and mucus contain antimicrobial enzymes, lysozyme, lactoferrin, that limit bacterial growth, but some microbes like Streptococcus mutans thrive in the oral cavity. Urine, normally sterile but can support microbial growth in conditions like urinary tract infections, UTIs, caused by E. coli.F. Inhibitory substances. Certain chemicals and antimicrobial agents affect microbial growth. Example, O antibiotics, such as penicillin, inhibit bacterial cell wall synthesis. O disinfectants, like bleach, kill microbes by disrupting cell membranes. O heavy metals, like mercury and silver, have antimicrobial properties. Growth of microbes. Definition: microbial growth refers to the increase in the number of cells rather than the size of individual cells. It occurs when microorganisms, such as bacteria, fungi, and algae, reproduce through cell division, leading to population expansion. Growth in bacteria. Bacteria grow mainly by binary fission, a process in which a single bacterial cell divides into two identical daughter cells. This results in an exponential increase in the bacterial population under optimal conditions. Phases of bacterial growth curve. When bacteria are grown in a closed system, batch culture, they follow a characteristic growth curve with 4 distinct phases. 1. Lag phase bacteria are adjusting to their new environment. No significant increase in cell number occurs. Cells synthesize enzymes and essential molecules needed for growth. Duration depends on environmental conditions and bacterial species. Second log, exponential, phase bacteria divide at a constant maximum rate. Population increases exponentially, 2, 4, 8, 16, etc. Nutrients are abundant and waste accumulation is minimal. The best phase for studying bacterial characteristics and antibiotic effectiveness. Third stationary phase. Growth rate slows as nutrients deplete and waste products accumulate. The number of new cells equals the number of dying cells, leading to a stable population. Some bacteria produce endospores, E. coli, Bacillus, Clostridium, or secondary metabolites like antibiotics. Fourth, death, decline. phase. Bacterial cells die due to lack of nutrients and toxic waste accumulation. Some cells survive longer by forming spores or resistant cells. Population size decreases significantly over time. Conclusion: understanding microbial growth and nutrition is crucial in medicine for controlling infections, optimizing antibiotic treatment, developing probiotics, and advancing diagnostics, vaccines, and antimicrobial resistance research.